Most human diseases are complex. They start and progress through mechanisms that typically involve many genes and proteins. Some of these molecules can be targeted. MicroRNAs are small RNA molecules in our bloodstream that can target protein levels by directly inhibiting protein synthesis or by promoting messenger RNA degradation. A single microRNA can target tens or even hundreds of messenger RNAs and proteins. Because of this, understanding these interactions holds great promise for finding novel ways to treat complex diseases. Treatment-resistant hypertension is a complex disease that can benefit greatly from research on microRNAs. In order to identify potential microRNA messenger RNA interactions that can be targeted for treating this disease, I built a predictive network model by integrating the values of more than 20 clinical variables with the expression levels of more than 2,000 microRNAs and messenger RNAs obtained from human blood samples. The model that I built is composed of individual decision trees, each of which is designed to predict the expression level of a single gene using all the remaining genes' expression levels as inputs. Each tree branch is split into two branches based on an expression threshold. Using data from 2/3 of the samples, these thresholds are optimized by creating the optimal separation between patient groups with significantly different gene expression levels. With the remaining 1/3 of the samples, model predictions are generated. When I use this approach for predicting the disease state, the Blue network module that was composed of several genes with similar network connectivity profile generated a predictive accuracy of 92%, suggesting that this network topology was highly specific to the disease state. Within this Blue module, T-cell receptor signaling pathway was the major biological pathway that was significantly enriched. This pathway is a key driver of inflammation that causes a great amount of kidney and vascular damage in hypertension patients. Among the 10 messenger RNAs participating in this pathway, ZAP70 turned out to be the one that was targeted by the highest number of microRNAs previously associated with cardiovascular disease in the literature. ZAP70 is a T-cell receptor-associated protein kinase that regulates the motility, adhesion, and cytokine expression of mature T cells that are key drivers of inflammation. In a recent study, mice lacking T cells were resistant against hypertension despite several experimental inputs that would normally trigger it. My finding, combined with what's known about ZAP70 in the literature, suggests that by targeting ZAP70 through microRNA replacement therapy and shutting down its activity, we can prevent adverse effects